Hi, this is Dr. Martin, and in this video, I'll talk about the antibodies and immune reactions. After watching this video, you will be able to understand the structure of immunoglobulins and describe the type of antibodies and their functions and the type of the antibody responses. And we will talk about the primary and secondary responses, immunotherapy, and define the classification of the immunity. Antibodies. Antibodies are soluble globular proteins that constitute the gamma globulin fraction of the plasma proteins. Each antibody molecule consists of 4 chains of amino acid, as you see in the picture. They linked by pairs of sulfur atoms that attract by disulfide bonds. The 4 chains form a Y-shaped structure. 2 of these amino acids chains are identical light chains, as you see light chains with the green color it showed, and the 2 are identical heavy chains. It shows with the purple as I showed. The heavy chains have about twice as many amino acids as the light chains. The 5 major types of antibody molecules are distinguished by a particular type of heavy chain. Most of the types of antibody molecules consist of a single Y-shaped structure, but some have as many as 5. Let's see this picture. As you see, the immunoglobulin M has 5 and immunoglobulin A has 2, but immunoglobulin G, E, and D has 1. The sequence of amino acid of the heavy and light chains confer the unique three-dimensional structure, in other words, conformation of each type of antibody, as in this case of other proteins. One end of each of the heavy and light chains consists of variable sequence. You will see here and here and here, that shows with the green colors. Okay, so those are the variable sequence of amino acid or variable regions. These regions are specialized to fit the shape of a specific antigen molecule, like a key and lock. Antibodies can bind to certain antigens because of the conformation of the variable regions. The remaining parts of the heavy and light chains are termed constant regions because their amino acid sequences are similar. Constant regions impart other properties of the antibody molecule, such as its ability to bond to cellular structures or to combine with certain chemicals. When you look here, you will see that the 80% of the immunoglobulins are immunoglobulin G type. The second most common is immunoglobulin A, and third most common is 6% immunoglobulin M. So when you consider the immunoglobulin G, A, and M, and actually conclude the majority of the antibody types. Immunoglobulin D and E is less than 1% each. Now let's look at the characteristics of major immunoglobulins. We said that 3 of the 5 major types of immunoglobulins include the most of the circulating antibodies. These types are immunoglobulin G, which accounts for about 80% of the antibodies, immunoglobulin A, which makes up the 13%, and immunoglobulin M, responsible for about 6%. And we said remainder of the antibodies are immunoglobulin D or immunoglobulin E. When we look to focus the immunoglobulin G is in the plasma and tissue fluids and is effective against bacteria, viruses, and toxins. Immunoglobulin G also activates the complement proteins. A newborn does not yet have its own antibodies, but has the immunoglobulin G that pass through the placenta from the mother that protect the infant against some illnesses to which the mother is immune. That's why when the baby is born, generally they do not get often sick. Until the maternal antibodies disappeared, uh, they disappear just about when the infant begins to make its own antibodies. Immunoglobulin A, and as you see, can be found in exocrine gland secretions. It is in tears, nasal fluid, gastric juice, intestinal juice, bile, and urine. A newborn receives the immunoglobulin A from the colostrum. It's a yellowish fluid that the mother's breast secrete for the first few days after giving birth, and then from breast milk. Antibodies in colostrum and breast milk protect against certain digestive and respiratory infection. In the colostrum, you will see the immunoglobulin G and immunoglobulin A concentrated. So it's important to give, especially a couple days of the life, giving your baby the colostrum. Immunoglobulin M is a type of antibody produced in plasma in response to contact with certain antigens in foods or bacteria. Examples of immunoglobulin M are the anti-A and anti-B antibodies. Immunoglobulin M also activates the complement. That means if you have a high immunoglobulin M, that means you are having an active exposure to a bacteria. You may be having the infection. Immunoglobulin D is on the surfaces of most B cells, especially those of infants. Immunoglobulin D acts as an antigen receptor and is important in activating the B cells. Immunoglobulin E appears in exocrine secretions with immunoglobulin A. It is associated with allergic responses. Antibodies respond to antigens in three different ways. First, methods of direct attack by antibodies. Second, activation of complement. Third, stimulation of local change, inflammation to help to prevent spread of infection. Let's talk about the first method and the direct attack by antibodies. In a direct attack, antibodies combine with antigens and cause them to clump. We call this agglutination. So agglutination is described as clumping of antigens, making phagocytosis easier. Or they may form insoluble substances. We call this what? Precipitation. Such actions make it easier for phagocytic cells to engulf the antigen-bearing pathogens and eliminate them. This naturalization, the effects, meaning that they cover the toxic portions of antigens, making them what? Harmless. Again, under the normal conditions, complement activation is more important in protecting against infection than is direct antibody attack. Okay. Now, activation of the complement. When certain immunoglobulin G or immunoglobulin M antibodies combine with antigens, they expose reactive sites on the antibody constant regions. This triggers a series of reactions leading to activation of complement proteins. Activated complement proteins coat the antigen-antibody complexes. We call this opsonization, and making the complexes more susceptible to the phagocytosis. Attract macrophages and neutrophils into the region. We call this chemotaxis. Agglutinate antigen-bearing cells, rupture cell membranes. We call this lysis, rupturing the cell membranes of pathogen. of foreign cells and neutralize the viruses by altering their molecular structure, making them harmless. We call this neutralization. Okay, let's do the next one. The third one is the stimulation of local change, inflammation, to help to prevent spread of infection. Immunoglobulin E promotes the inflammation, maybe so intense that it damages the tissues. This antibody is usually attached to the membranes of widely distributed mast cells. When antigens combine with the antibodies, resulting antigen-antibody complexes stimulate the mast cells to release the biochemicals such as histamine that cause the changes associated with inflammation, such as vasodilation and edema. Okay, let's understand what is immunotherapy is. Immunotherapy makes the use of immune system components to fight disease. And when you use the immunotherapy, you can use the humoral immune response, giving the patient the antibodies, or cellular immune response, giving the patient what? Cytokines. When we look to antibodies, we call them monoclonal antibodies because a specific B cell produces a single type of antibody. It's monoclonal, one type. Can be fused with a cancerous cell to produce a cell that produces one type of antibody and keeps dividing. Used in research, agriculture, pregnancy tests, detecting new or reoccurring cancers, delivering medications, or specifically to what? The tumor, to deliver the specific the medications. Cytokines is the part of the immunotherapy. It can be produced in large quantities due to recombinant DNA and monoclonal antibody techniques used to treat the leukemia, multiple sclerosis, hepatitis, genital warts, melanoma, and kidney cancer. When we look at the immune responses, we have two types, either primary or secondary. So when we look at the immune responses, with primary immune response occurs when B cells and T cells become activated after the first encountering the antigens for which they are specialized to react. During this response, plasma cells release antibodies, immunoglobulin M followed by immunoglobulin G. So first immunoglobulin M, then immunoglobulin G. The antibodies are carried by the blood throughout the body where they help destroy antigen-bearing agents. Production and release of antibodies continues for several weeks, maintaining higher numbers of antibodies in the plasma. And after a primary immune response, some of the B cells produced during proliferation of the clone will serve as a memory cells. Let's go to the next one. And if the same type of antigen is encountered in the future, the clones of these memory cells enlarge and they can respond rapidly with immunoglobulin G. These memory B cells, along with the memory T cells, produce a secondary immune response or an amnestic response. In lymph nodes, follicular dendritic cells may help the memory by harboring and slowly releasing the viral antigens after an initial infection. This exposure constantly stimulates the memory B cells, which present the antigens to memory T cells, maintaining the immunity. Memory B cells live for many years, but antibodies remain for months or years. Now, when we do the practical classification of immunity, it can be either naturally acquired or artificially acquired. If it's naturally acquired, that means obtained by a natural process, such as getting or recovering from a disease, or the mother gave the fetus or infant. So we call these naturally acquired. But when we say artificially acquired, that means it requires an injection instead of a natural process. So it can be either naturally acquired or artificially acquired. And at the same time, classification of immunity based on whether it is temporary or permanent. And if it's temporary, we call this passive immunity. A temporary immunity obtained via antibodies. No antigen exposure, no immune response is evoked in the person's immune system. You already give the person already prepared antibodies. For example, if there is a snake poisoning or scorpion poisoning, you give the antitoxin. You already giving the person already prepared antibodies. That is a artificially acquired passive immunity. An active immunity, meaning permanent immunity, obtained via antigen contact. Immune response is evoked and memory B cells are produced by that particular person. In summary, humoral immunity has two types, either active or passive. If it's active, it can be either naturally acquired, good example having the infection, contact with pathogen, and artificially acquired, either vaccines can be dead or attenuated, that means weakened and pathogens, but you created your own antibodies. The passive type of humoral immunity, it can be either naturally or artificially acquired. If naturally, antibodies pass from mother to fetus is natural via placenta or the infant in mother's milk. Artificially acquired passive type of humoral immunity, injection of immune serum, especially the gamma globulin, like I gave the example, the scorpion toxin, antitoxin, or snake antitoxin. I hope you enjoy it, and please feel free to click the subscribe button to watch more videos from me.